morning. Welcome to our online event, Environmental Risks with Parkinson's. Today's program is brought to you by Parkinson's Community Los Angeles. Today we are so fortunate to have with us Dr. Beate Ritz, MD, Ph.D. I'm Angela Neff, PCLA board member. For those who don't know us, we are a nonprofit that supports families living with Parkinson's through free education programs like this one, online and in-person support groups, social events, and more. Today's conversation is brought to you by our generous sponsors, Abbott, Boston Scientific, Kyowa Kirin, Medtronic, and Supernus, and by important donations from people just like you in the Parkinson's community. If you appreciate our work, please consider making a donation at PCLA.org. Your donation helps us continue this really important work. Please join me in welcoming the wonderful, the fabulous Dr. Ritz. Dr. Beate Ritz is a professor of Epidemiology and Environmental Health Sciences and Neurology at UCLA. Her research focuses on environmental and genetic factors that influence the risk of Parkinson's disease. She is the principal investigator of two of the largest community-based studies of Parkinson's disease worldwide, located in the Central Valley of California and in Denmark. These studies identified environmental risk factors for Parkinson's disease, onset, and progression. Most recently, her lab has started to integrate the gut microbiome into these investigations. Thank you so much, Dr. Ritz.
Thank you very much for this wonderful introduction, Angela. And I think I'll just take it from here. I have to make a few disclosures, though, because I am involved in some litigation on Paraquat exposure for patients who are taking Syngenta to the courts. So I'm an expert, not for any particular person, but for the science behind pesticides, specifically Paraquat and Parkinson's. And otherwise, I receive all of my research funding from the National Institute of Health, some from the Department of Defense, Michael J. Fox, American Parkinson's Disease Association, and the Parkinson's Foundation. And I'm very grateful to all of the funders who have basically helped me do this research over the last 25 years. So I'm talking today to you about pesticides, and the question is why? Well, our food production actually depends on pesticides, as well as the public health sector. In terms of malaria and Zika control, you will find insecticides have an important role. And we have, of course, commercial as well as private applicators who protect necessary crops, but also roadsides and golf courses from insects and weeds. And therefore, we have quite widespread use of pesticides. We would find them near homes. We find them near residences who are in rural areas or near crops. And the reason for that would be that they drift from an application site into those homes. But there might also be some take-home contamination if somebody's actually working on fields and gets their clothes contaminated, or otherwise brings home something that contained containers that contained pesticides. Most people, however, would not live in close proximity to agriculture. Quite a few, I think, live actually near golf courses or roadsides where also pesticides might be used. But we are also using indoor and outdoor pesticides on lawns and in gardens. And in fact, one survey showed that 85% of all US households are actually storing at least one pesticide at home. And in the dietary and Haines biomonitoring, that's a national survey of Americans, they find that in the urine you can find dietary contaminants from dietary sources in just about everybody's urine. Even so, what they find might actually change over time. Pesticide production, because of its usefulness for so many reasons, has industrially increased from the 1950s to the common year. Actually, I'm showing you on the left just up to the year 2000 here. But if you're looking at this slide from a German foundation, they are showing you that currently Latin America uses the most growers in Latin America, followed by China and the US and then Europe. And that was in 2022. And this kind of use is not diminishing. It is, as this slide indicates, actually increasing because of many reasons, but it's not going away. So pesticide use worldwide is not going away. US-wide survey and Haines keeps finding pesticides in our urines. It was 96% of all samples had an organophosphate chlorpyrifos in the 2000s. And that survey in 2015, 2016 then found that now it's actually pyrethroids. Half of the US population now shows pyrethroid pesticides in their urine. Why do we look at Parkinson's and pesticides? Well, this is actually going back to the early 1990s that people scratched their head and said, well, is there something with pesticides and Parkinson's going on? And yes, there were even a lot of studies who asked people that they enrolled in a small case control study on all sorts of risk factors. Have you ever used pesticides? And then basically sometimes also asked, what have you used? And on the right here, you see all these dots that are above this line indicate that these studies found positive associations with pesticide use. And overall, they estimated that it's about 90% to twofold risk increase if somebody had reported the use. Well, but that's just pesticides in general. We are, however, much more interested in identifying bad actors, bad acting pesticides that are neurotoxic, that harm us, than just indicting every single pesticide on the planet because they won't go away, right? So we might as well find the worst offenders, and hopefully we can do something to eliminate exposure to them. And this goes back to Paraquat, which I mentioned earlier, because Paraquat looks very similar to a known dopaminergic toxin called MPP+, which is the metabolite of MPTP. This is Paraquat. MPP+ just lacks this one H3C and plus group on this side. Otherwise, they're chemically identical. Because of this similarity, Paraquat has been interested researchers for quite a while in terms of is it or not causing Parkinson's disease. And you can see in California alone, it's not like we have diminished its use. It's actually been going up since 1980s to 2020, and it keeps going up, the use. And that is, even so, the European Union, even Brazil, have outlawed or banned Paraquat use in the past. But the US is still using it because it's a very effective herbicide. California is still using it. So back to my study that was introduced by Angela so nicely before. It's called the Parkinson's Environment and Genes Study, and it's a case control community-based study in 3 rural counties north of us, basically Kern, Tulare, and Fresno counties that you see on the map here. And since the year 2000, we have worked with neurologists and clinics in this area. And now the California Parkinson's Disease Registry, that's California-wide, but we are getting cases from these areas reported to us. And we've been reaching out to these individuals and enrolled and have seen, our movement disorder specialist, Dr. Bronstein, has seen about 850 patients. And we also enrolled about 1,000 controls. These are the people on whom most of the results that I'm going to share with you are based on. And they have been incredible. We are so indebted to these individuals because they spend almost a whole day with us in exams and with questionnaires and sharing with us their blood, their saliva, their feces, their urine, anything we ask them for. It's a big deal because they have to make the effort to see us and to get us these samples. So thank you, thank you, thank you. So the first thing we did is something that every Parkinson's study basically does. We are looking at occupational pesticide exposure, which is reported to us in these questionnaires. And we have asked every individual whether they've ever occupationally, because this is a highly agricultural region, used pesticides. And you can see that those who answered yes had about a 2.5-fold risk increase. So definitely occupational use increased risk. And when we look at specific agents, you can see it looks like fungicides are a little worse than herbicides or insecticides, but that's occupational use data. We also asked these individuals whether they've ever used pesticides in their households. And we also asked them what type of pesticide and for what use. Was it for herbs, for weeds? Was it in their households for insects? And what type of can label did they have in their household? And did they use frequently? And when you looked here, any use had about a 47% risk increase. But when you reported that you had organophosphate pesticide use, it was even a bit bigger. It was 1.7, so 70% risk increase. And this kind of confidence interval tells you, yes, it was statistically significant. But it was self-reported use, no more at this point. So why we got this money from so many different agencies was because we have something in California nobody else has. Mandated by law, we have agricultural pesticide use reporting, where we find out in what county and what 1 square mile location, at what date, on what type of commodity, and how a pesticide was applied. And you can see here, these are different ways of applying. Here's a tractor spraying an orchard. Here's a tractor spraying a field. But here you have that little yellow plane flying over, and that's crop dusting. And you see buildings down here, and those could be farms, but they could also be schools, or they could be homes in rural environments. And when this wafts over, you can imagine that definitely a little bit of what they spray here might get into those homes. So what we are doing with all of the data we collected is we asked people to go back to the 1970s and give us their address, and they did. And then we plotted these addresses on a map here. And after using this kind of computerized system where we have the use of the land use, meaning, is this, for example, grapes? Is this an orchard? Is this a field? And what was applied on these? And then overlapping, you can see this is how pesticides are applied, in this case, on grapes. And we are then summing all of this up over 30, 40 years of addresses that somebody is giving us. And we can tell you how much poundage of a specific chemical was applied within a 500-meter or 1,000-meter buffer, which is about a quarter mile from your home in these areas. So does this system, however, tell us whether somebody was really exposed? Well, the EPA did some monitoring in a small community called Parlier on the top of elementary schools. And here you can see these flags. They are the monitoring instruments, and they monitored all year. And you can see here what the pesticide use reporting system showed you in terms of one specific pesticide called diazinon being applied within this 8-mile distance from this community. And here they are monitoring, here they are applying. Over here you see a map, or better, a graph, that shows what was applied, which is this kind of pinkish. So these are applications according to the PUR system, and then what was detected on the roofs of these schools. And you can see that detection and what was applied is basically identical. So I think we can actually use this system to at least find out what's in the air once these applications happen. This is our first study we ever published with the data in 2009. So it took us 9 years before we could even publish the first paper. And that was on Paraquat use and Maneb, which is a fungicide. And I mentioned before that fungicides also showed up in the occupational data as being problematic. And here, all I want to show you is that we estimated an about 75% increased risk using this pesticide use report data for residential and occupational addresses when these individuals, or better, the patients, were exposed to both Paraquat and Maneb. We've been criticized after that, we also looked at Maneb. At the time, an animal study had suggested that these two agents together are much worse than one of them. So we have now, with a lot more data, written another manuscript with double the number of cases, double the number of controls. And that manuscript is only on Paraquat and Parkinson's disease, and hopefully will soon be out in press. But right now I can't tell you about it because it's still under review. But it's hopefully coming, and you can see what we see for Paraquat only. So humans definitely encounter more than one pesticide in their life, definitely more than Paraquat. And my colleague, Dr. Kimberly Paul, has said, well, why don't we just look at every pesticide that we can find within that 500-meter buffer zone around a patient's home or a control's home and see whether they all contribute to Parkinson's. And these are all the pesticides she fished out from that screen. And we call it a pesticide-wide exposure study, and it has been published in a journal recently. And basically, you can see that all of these red dots show increased risks for Parkinson's disease. On this side, you can see that there are down here is all the names of these pesticides, and they should also go on this side. Basically, what the red is showing you is that there's high correlation among groups of pesticides that seem to be applied together. And that is a problem because in epidemiology, you cannot really disentangle these effects. So we went with colleagues to DOD, Dr. Kurana and Dr. Rubin at Harvard, and said, can you help us test which of these pesticides might be the worst actor? And they have human pluripotent stem cells that they derived from alpha-synuclein carrying patients and that they're using for this kind of testing. So we asked them to test 70 out of these 200 pesticides that we had tested, the 70 that we thought might increase risk. And here you can see they fished out these different pesticides, all the ones that cause harm to the cells so that the viability, the number of cells goes down, and that's what you see here. These are all the pesticides that kill dopaminergic neurons that are made from pluripotent stem cells of Parkinson's patients with synuclein mutations. So we are now thinking that, these might be the especially bad actors, even though this is just one screen we can use to test for toxicity. And we also did go back to our own data and said, well, we know these pesticides cluster. Let's look at how they cluster. Here we have something called our cotton cluster. These are all the pesticides applied on cotton. And what our colleagues, Dr. Królewski in Dr. Rubin's lab did is he used mixtures or combinations of these pesticides on these same cells and looked again how much they did not survive, right? So the survival is up here, and when it goes down, these cells have been dying. And you can see that there are some pesticides that are always in this right hand of the curve, meaning they cause a lot of cells to die. And one of these toxicants is trifluralin, which in combination with these others seems to be specifically toxic. And trifluralin had also been picked out by another epidemiologic study, a large study of agricultural workers before. So regulatory agencies rule on specific chemicals, not on mixtures. That's why we always have to go back to, is this chemical a problem? And we have been using this so-called gene environment approach to make sense of whether or not we can say more about these pesticides. And this is work we've done over the last 20 years, again and again. And we have done it for different kinds of genes and different kinds of systems. And what you're seeing here are not Parkinson's genes, but genes that help repair in cells DNA that are scavenging toxins, that are metabolizing pesticides, or helping pesticides to enter cells like these two. And based on these kind of genes that actually make it difficult for pesticides, for example, to be detoxified, we did what we call a gene environment interaction study for organophosphate pesticides. So you have an organophosphate pesticide like chlorpyrifos that was shown to be in everybody's urine basically in the 2000s. And you go down this toxic detoxification pathway and you see PON1, the paraoxonase. And that is an enzyme that actually shows a 40-fold difference from possibly you and your neighbor. There are those people who are very fast metabolizers and those who cannot get rid of this pesticide very fast. They're called slow metabolizers. And interestingly, it's genetics that determines whether you're a fast or a slow metabolizer. And down here is the genetic information you would need to know whether you're slow. And we used exactly that in our patients and control to find out whether people have been slow or fast metabolizers. So in this slide, you can see that if you're exposed to these OP pesticides, diazinon, chlorpyrifos, or parathion, only if you're exposed and you're also a low or slow metabolizer, are you at this very high risk of PD. So a 2.5-fold, almost 3.2-fold, and a 3-fold risk increase you have when you have no capacity to get rid of these pesticides. And when you are having a medium capacity, you are at increased risk, but not as high, okay? Now, this is based on our PUR system, whether you live near these agricultural fields that have been sprayed. But what we also did is we looked at our household OP pesticides here, and we determined who has been using one of these pesticides in their homes frequently and is also a slow metabolizer. And here you can see, even if you use these pesticides a lot, you're not at an increased risk, really. However, if you are not a slow metabolizer. However, if you are a slow metabolizer, that's really what puts you at that 2.6-fold risk increase for PD. So it's not the gene, it's not that you're slow or fast, it's not whether or not you have used these pesticides, it's the combination of you not being able to get rid of them and being exposed. And we played this game with a few other pesticides, the dopamine transporter that is possibly transporting the pesticide into your cells. And you can see this is from a data that a colleague gave us, where they found a 5-fold risk increase if that dopamine transporter acts faster. And we were able to replicate that in our study as well. And we did that with transporters that help agents cross the blood-brain barrier. Again, you're at much higher risk from the exposure to a certain pesticide, in this case, the organochlorine pesticides, in my colleague's study, as well as in our own study, when you're exposed. So genetics come together with the exposure. And we've done that ourselves also. My colleague Jeff Bronstein has done that for the aldehyde dehydrogenase gene that detoxifies dopal to dopac, and therefore makes this pathway be much less toxic. And when we look at that gene that codes for this enzyme, you can see, again, with pesticide exposure, that's what puts you at higher risk if it's a pesticide that actually interacts with this kind of genetic mechanism. We also looked at immune system, and again, we found the same story. If you have a highly reactive immune system and you're exposed to a pesticide that affects the immune system, that's when you're at high risk. And the same thing for nucleotide excision repair, which is a DNA repair. If you have mitochondria inhibitor exposure and you cannot repair the breakage, that's when you're at higher risk. And all of this is published and you can find in the literature. We also then went and said, well, gene environment, what about the metabolites that you can find in patients? And this is what one of my students, Chian, did. He looked at combinations of pesticide exposures, those organophosphates, pyrethroids, and organochlorines, and what kind of signature you can find among patients who have been highly exposed, or among humans who have been highly exposed, in this case, whether patients or not, to these kind of pesticides. Here, he's just looking at people who are exposed, and these are all the metabolome pathways that he picked out, and he put them in this nice picture for us to show that what he picked out, that these pesticides are affecting when you are exposed, are pathways related to mitochondria, pathways related to oxidative stress and to inflammation. And all of these 3 pathways have been linked to Parkinson's disease before. Now, before I finish, I want to briefly introduce this new concept of the gut-brain axis and the microbiome. And that's basically what we are going after now in the Central Valley, collecting stool samples from these individuals. And that is based on the hypothesis that our non-motor system, especially the gastrointestinal non-motor symptoms that many of the Parkinson's patients are having years and some even decades prior to the motor symptoms before they are diagnosed, that they actually are related to something that's happening in the gut and possibly contributing to alpha-synuclein being retrograde transported into the brain, so from the gut into the brain through the vagal autonomous nervous system, and then make the protein in the brain misfold. And this hypothesis has been supported by animal models, but also by some studies in Denmark, where investigators looked at people who had their vagus nerve cut. That was a kind of treatment done for ulcers for many, many years, and what their risk of Parkinson's was, and found that they indeed, the ones who had their vagus nerve cut, and the parts of the vagus nerve, especially that innervated the colon, were at a lower risk of Parkinson's. So there might be something that is transported from the gut to the brain that's toxic, and possibly it's alpha-synuclein. So we are currently collecting fecal samples, but we had already done some preliminary analyses. And here I'm showing you a paper of pyrethroid pesticides, which are common right now for killing insects. And this is a paper where we show that these pesticides actually have been showing links to depression among our patients and our controls. And over here, I'm showing you the kind of microbes that you find changed with exposure to pyrethroids. And I just want to show you there are these microbes in the gut, in the fecal samples that we analyzed, Citrella, Haemophilus, and Prevotella, that are found less in the exposed, the pyrethroid exposed individuals that we have samples from. And if you go to a systematic review on what the gut microbiome looks like in patients that are anxious and depressed, these are the bacteria that are reduced. Citrella reduced, Prevotella reduced, Haemophilus reduced in most of the studies out there. And these are exactly the microbes we find reduced with pyrethroid exposure. And before we had shown that pyrethroid exposure actually might have something to do with depression in our study. So I think that's a nice proof of concept that we are on the right track to seeing something useful with the fecal samples. This is preliminary data. This is actually not published yet, this slide. We are still collecting samples and we will reanalyze them. This is a study we are now having in review about the healthy diet and the PD microbiome. So we are going from diet to the brain through the gut microbiome. And what I want to show you here, and then leave you to ask me questions, is that these are all bacteria that have been shown to be more in lower abundance with PD. And these are the ones that have been shown to be in higher abundance with PD compared to controls in our data as well, but mostly in the literature. And when you have a very good, a high HER, healthy eating index, so a very, very good diet, you have less of the good bacteria, and you have of the anti-inflammatory good bacteria, and you might actually have more of the ones that are not good for you. And what specifically contributes? Well, it's high fiber that contributes to a good diet, and a lot of sugar that's not fiber content, but sugar does the opposite. So that's what we're seeing right now. Again, this paper is not published yet. You can't quote it yet, but we are trying to get it published. Why is that pesticide study so important? Well, because we have to influence regulation with a really good scientific story. If we are not doing good science, we probably will not influence public policy. And I want to really emphasize that different from pharmaceuticals that are tested in the test tube and registered and approved by the FDA, and later also have to be followed up for side effects, unintended side effects, the pesticide industry goes from testing to registration and approval, but never is asked to actually look at post-approval monitoring of human health effects. So what we call pharmacovigilance, which is that looking for side effects after market approval and introduction into the market, is not happening currently with pesticides. And I think it's time that we do this. This was an editorial in Science magazine that actually asked for that. This is my unsung hero, Mark Siegel, who was the first advocate for the PD registry in California. And I'm thanking I'm thanking Arnold Schwarzenegger here for supporting the registry in 2004, but it unfortunately took us until 2017 to get it funded by the state. We hope it will keep being funded. And these are us lobbying for this, activists and scientists going up to Sacramento in order to get this funding finally in place. Here we are in 2015. And I really want to emphasize Greg Wasson was very important, as well as Mark Siegel, for this effort. And Greg passed away recently. So thank you, Greg. And these are all of my colleagues and students who worked over most of 25 years with me, coming and going, the students, staying with me the whole time, Dr. Bronstein and some of his fellows. And I especially want to thank Dr. Sinsheimer, who passed away this year and was the statistician on this study for the last 25 years. And of course, all of the funders and the families of the patients who help bring the patients to our exam rooms and who encourage them and help them all the way. Thank you so much for participating.
Thank you, Dr. Ritz. That was incredible. We're going to move to questions, speaking of questions now. So one question was, for people who want to do everything they can do to detoxify themselves regularly, do you recommend regular sweating, exercise, sauna, antioxidant-rich foods, that sort of thing? Like, do you have any opinions on those sorts of things? How to treat?
Yeah. Detoxification is a different word from what I've been using in my slides. So when I talked about detoxification, it was really how your enzymes, mostly in the liver, but even your microbiome, which is basically a second liver, metabolizes potential toxins or generates potential toxins. So what I was referring to with the pesticides were all sorts of pathways where the handling of the pesticide is not as good in certain people as in others. Who these people are, we sometimes can guess because there's certain genes that, are really active in helping with that. But basically what you're referring to as sweating and detoxification may be too late because the agent has already been in your system. Definitely you want to keep a healthy microbiome because that can actually possibly detoxify or not allow toxin to be generated before they get into your system and through your blood or through your or through other means and ways get into your brain eventually. So keeping your microbes in your gut healthy and keeping them happy, I think, is probably a really good idea. And that's why I, in the end, put this slide with the healthy eating index. And healthy eating is basically lots of greens and lots of fruits. And yes, I know they might be having pesticides, so you might think about that, but they also help you detoxify.
Great. And what about saunas? There's a lot of talk about, ice baths and saunas, going from ice to hot, really cold to hot. Do you have any opinions about that sort of thing?
I love both, but I have no scientific opinion whether they work. If they make you feel good, do it.
Yeah, I think that at the very least they activate our dopamine.
Exactly.
How can a person determine, if there is a way, whether they are a fast or a slow metabolizer? Do they need to go through your diagnostic process?
Yeah, I would not recommend trying that because there are so many different genes that contribute to this, and you might find, yes, I'm good at detoxifying organophosphates, but maybe you're not so good at detoxifying pyrethroids or neonicotinoids, or whatever next pesticide might be introduced into the market. So you might actually have a false sense of, oh, I'm fine because I can detoxify organophosphates really well. But, the exposure is already something else. My public health approach is let's limit exposure for everybody as much as possible, rather than trying to figure out whether I'm superwoman who can detoxify.
Monica Gross of the Poise project with the Alexander technique asks, can you go over again about the impact of an individual's genetics around their dopamine transporters as opposed to the slow and fast metabolizers that you spoke of?
Yeah, so I went over that really fast because I wanted to allow for questions. Basically, the dopamine transporter has been a so-called candidate gene for transporting not only dopamine back into the synapse, but possibly other toxic substances like paraquat, because they are available as well if you're exposed. And we know that paraquat needs to be transported into cells. It doesn't get there on its own. And the dopamine transporter has been one of the hypotheses of, okay, if that dopamine transporter is really good at it, it also transports some toxins into the synapse, and then the toxins wreak havoc. And that was the idea behind this gene-environment interaction. It looks like something is up with the dopamine transporter genetically that makes us vulnerable to these two pesticides I checked into, Paraquat and Maneb, but we don't really know what it is. So it's hard to say what it is about the dopamine transporter that made these synapses more vulnerable to the pesticides, or the body more vulnerable. Could be very complex. But with the detoxifying, metabolizing enzymes, that's very easy to understand, because they are basically just getting rid of the toxin or generating a toxin at a fast or a slow pace. And they are easier to understand mechanistically because of that.
Similar question. Are we any closer to determining whether pesticides or something else was the primary cause of one's Parkinson's diagnosis?
Yeah, that's the $3 million question. We all would like to know what caused our Parkinson's, your Parkinson's, but generally what I'm telling patients or anybody who asks me is it's probably not just one thing, it's probably a combination of things that have happened, but there might be a preponderance of one over the other depending on your exposure profile. So you might have been the person who lived near these fields or who ingested a lot of pesticides, and you might have been unlucky enough to be vulnerable because of genetics, because of other issues, not just genetics, and you got Parkinson's because of that. Whether it was one pesticide or another, or mostly pesticides, for an individual, we will probably never know because the pesticide might be long gone from your body, right? We won't find it anymore. Right. It goes in, it harms your dopaminergic system, it goes out. We don't find it anymore. So we can only guess at that point.
Yeah, I've heard several different people talk about having it in their families, having Parkinson's in their families, and feel their strong conviction that that was caused from the fields that they lived around with, that had a lot of exposure to these pesticides. Yes,
so that's definitely one hypothesis. You might be, or your family might be, vulnerable, and that's why multiple individuals get it, right? And the exposure was there because you lived in one place.
The whole thing about the slow processing, people who have to me that makes so much sense, because many people think, why me? I exercise, I eat well, I do this, I do that. These other people I know that lived in the same environment, trash their bodies, and they don't have Parkinson's. But it makes sense that it's something biological that where you've just processed the ability to defend yourself against these pesticides is reduced in some people for whatever reason. We don't know at this point. We don't know why. We just know that in fact there are differences. Am I understanding that correctly?
You're absolutely understanding this correctly, but I want to emphasize it doesn't only have to be genetics. It can also be that multiple exposures come together. For example, we have another paper that I didn't mention where we show that if you have been Paraquat exposed and you also had a head injury that caused a 5-minute unconsciousness, then you are actually at a much higher risk than when you only had a head injury or only were pesticide exposed. And this is probably, so that's one study we published, but this is probably also true for a lot of other agents. We saw signs that lead exposure long time ago, like 30 years ago, lead exposure puts you at risk. So what if you are lead exposed and then you're also exposed to pesticides and you also have a head injury, right? It kind of accumulates over time.
Got it. And kind of building on that question, how different is the data for people with PD and exposure to pesticides in urban areas versus farming areas?
Right. So the reason why our controls come from farming areas is because you want to not mix up what exposes people in urban areas with what people in rural areas have. You want to have the same baseline, right? Hopefully.
Yeah.
So, but in urban areas, we have lawns and, we have pesticide applications to certain roadways and to certain types of parks or to golf courses. So that's one way how we can get pesticide exposed. However, what I'm most worried about, and I didn't talk about today, is air pollution. And we actually have a study just recently published showing that, traffic-related air pollution, as well as other types of particulate matter air pollution, likely also metals that are in this kind of air pollution, are quite harmful as well and put you at increased risk for Parkinson's as well. So we have to clean up our air, no matter whether it's air pollution from industry, from cars, or whether it's farms.
Absolutely. Somebody asked a question about Greg Wasson that you mentioned passed away. And they just wanted to confirm that and also wanted to know if Jeff's wife was still living.
I believe, but I don't know. I actually only found out about this from the Barcelona meeting.
Oh, okay. It's definitely a huge question and scary. This whole thing about the Agricultural Labeling Uniformity Act, that there's, do you want to talk about that?
Yeah. I think generally labeling yes or no gives you a choice or gives you information, but from a public health perspective, you want to avoid exposures. You don't want to just label something and say, no, it's up to you to be safe or, stay away from fields or this or that. I think we really need to have best practices in using pesticides, in having certain distances of applications from homes and residences, in making products we use in our homes as non-toxic as possible, and also keeping our air clean. So I believe we should not go to labeling helps avoid exposure. I don't think it will. But yes, it's better than not having any information, that for sure.
Okay. My impression from this was that the concern was that the corporations are trying to get themselves protected so that they don't have to.
Yes. Can I interject for a second?
Yeah,
Dr. Ritz, I've been following, just kind of food and nutrition and kind of where farming is headed in this country. And this particular bill was just introduced, I think, like a month ago, and there's a bunch of pesticide companies and farmers, conventional farmers, that have signed on to support it. And basically what it does is it keeps pesticide companies from being sued. It's labeled
Yes.
a labeling act, but it's a tricky label.
Yes.
So anyway, I'm not sure how much you know about it. It's kind of new, and it just came on my radar, and I was curious what your thoughts
I don't know. Yeah, thank you for introducing that. I didn't know about much of it, but my general opinion is labeling is better than not. But if that doesn't protect the public, it's not what we should push for.
Mark Ikimura would like to know if when farmland is being converted to residential housing, are these areas required to detoxify the land before they build on the property? Do you know anything about that?
Really good question. The biggest problem would be with the pesticides that don't disintegrate. Those are mostly the organochlorine pesticides, like DDT. They are still being found in soil, but I don't believe that there's anything that says this is like a brownfield or a toxic waste site, and we need to take care of what the topsoil looks like in terms of contaminants. I have not seen any movement in that direction. So putting a house on top of it, it probably, I mean, if you have a foundation and you have concrete everywhere, you might not get in touch with the soil, but if you have a garden or a yard, yes, it might be a problem.
Thank you so much. And I know you have a meeting to get to, so we're going to wrap it up. Thank you, I knew this was going to be incredible. We could have you on 3 more times and have more questions, I'm sure. But today's event was made possible by our sponsors, Abbott, Boston Scientific, Kiowa Kirin, Medtronic, and Supernus. We have some wonderful events coming up, which I'll tell you about in a moment. But if you enjoyed today's program, please visit PCLA.org. Make your donation today. By donating to PCLA, you can join us in our mission to improve the lives of families in our community living with Parkinson's. You can find our contact information on the bottom of this screen. PCLA is a nonprofit. All donations are tax deductible. And we have some great upcoming events. On Thursday, November 30th, the latest research from the TAS Lab, Dr. Peter A. TAS. And don't miss our annual conference, Life Beyond the Basics, on Saturday, December 2nd, with PCLA's Medical Advisory Board, Dr. Wael El-Nashif, Zenos Mason, Nicholas Sumsky, Adrian Keener, Michelle Tagliotti, Melita Petrosian, and Jeffrey Wertheimer. Links to register for these events, as well as updates for all of our programs, will be sent out to our email list, or you can visit PCLA.org. Thank you, everyone. This was incredible. Thanks for the great questions. Have a wonderful, wonderful day.